Section 48 of Gray's Anatomy, Part 4. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Anatomy of the Human Body, Part 4, by Henry Gray. The organ of smell. Organon olfactorius, the nose. The peripheral olfactory organ, or organ of smell, consists of two parts: an outer, the external nose, which projects from the center of the face, and an internal, the nasal cavity, which is divided by a septum into right and left nasal chambers. The external nose, nasus externus, outer nose. The external nose is pyramidal in form, and its upper angle or root is connected directly with the forehead. Its free angle is termed the apex. Its base is perforated by two elliptical orifices, the nares, separated from each other by an antero-posterior septum, the columna. The margins of the nares are provided with a number of stiff hairs, or vibrissae, which arrest the passage of foreign substances carried with the current of air intended for respiration. The lateral surfaces of the nose form, by their union in the middle line, the dorsum nasi, the direction of which varies considerably in different individuals. The upper part of the dorsum is supported by the nasal bones, and is named the bridge. The lateral surface ends below in a rounded eminence, the ala nasi. Structure. The framework of the external nose is composed of bones and cartilages. It is covered by the integument and lined by mucous membrane. The bony framework occupies the upper part of the organ. It consists of the nasal bones and the frontal processes of the maxillae. The cartilaginous framework, cartilaginous nasi, consists of 5 large pieces, namely the cartilage of the septum, the 2 lateral and the 2 greater alar cartilages, and several smaller pieces, the lesser alar cartilages. The various cartilages are connected to each other and to the bones by a tough fibrous membrane. The cartilage of the septum, cartilagio septi nasi, is somewhat quadrilateral in form, thicker at its margins than at its center, and completes the separation between the nasal cavities in front. Its anterior margin, thickest above, is connected with the nasal bones, and is continuous with the anterior margins of the lateral cartilages. Below it is connected to the medial crura of the greater alar cartilages by fibrous tissue. Its posterior margin is connected with the perpendicular plate of the ethmoid, its inferior margin with the vomer and the palatine processes of the maxillae. It may be prolonged backward, especially in children, as a narrow process, the sphenoidal process, for some distance between the vomer and the perpendicular plate of the ethmoid. The septal cartilage does not reach as far as the lowest part of the nasal septum. This is formed by the medial crura of the greater alar cartilages and by the skin. It is freely movable, and hence is termed the septum mobile nasi. The lateral cartilage, cartilagio nasi lateralis, upper lateral cartilage, is situated below the inferior margin of the nasal bone, and is flattened and triangular in shape. Its anterior margin is thicker than the posterior, and is continuous above with the cartilage of the septum, but separated from it below by a narrow fissure. Its superior margin is attached to the nasal bone and the frontal process of the maxilla. Its inferior margin is connected by fibrous tissue with the greater alar cartilage. The greater alar cartilage, cartilagio alaris major, lower lateral cartilage, is a thin, flexible plate, situated immediately below the preceding, and bent upon itself in such a manner as to form the medial and lateral walls of the nares of its own side. The portion which forms the medial wall, crus mediali, is loosely connected with the corresponding portion of the opposite cartilage, the two forming, together with the thickened integument and subadjacent tissue, the septum mobile nasi. The part which forms the lateral wall, crus laterali, is curved to correspond with the ala of the nose. It is oval and flattened, narrow behind, where it is connected with the frontal process of the maxilla by a tough fibrous membrane, in which are found 3 or 4 small cartilaginous plates, the lesser alar cartilages, cartilaginous alaris minoris, sesamoid cartilages. Above it is connected by fibrous tissue to the lateral cartilage and front part of the cartilage of the septum. Below it falls short of the margin of the nares. the ala being completed by fatty and fibrous tissue covered by skin. In front, the greater alar cartilages are separated by a notch, which corresponds with the apex of the nose. The muscles acting on the external nose have been described in the section on myology. The integument of the dorsum and sides of the nose is thin, and loosely connected with the subadjacent parts, but over the tip and alae it is thicker and more firmly adherent, and is furnished with a larger number of sebaceous follicles, the orifices of which are usually very distinct. The arteries of the external nose are the alar and septal branches of the external maxillary, which supply the alae and septum, the dorsum and sides being supplied from the dorsal nasal branch of the ophthalmic and the infraorbital branch of the internal maxillary. The veins end in the anterior facial and ophthalmic veins. The nerves for the muscles of the nose are derived from the facial, while the skin receives branches from the infratrochlear and nasociliary branches of the ophthalmic and from the infraorbital of the maxillary. The nasal cavity, cavum nasi, nasal fossa. The nasal chambers are situated one on either side of the median plane. They open in front through the nares, and communicate behind through the choanae with the nasal part of the pharynx. The nares are somewhat pear-shaped apertures, each measuring about 2.5 centimeters antero-posteriorly and 1.25 centimeters transversely at its widest part. The choanae are two oval openings, each measuring 2.5 centimeters in the vertical and 1.25 centimeters in the transverse direction in a well-developed adult skull. For the description of the bony boundaries of the nasal cavity, see pages 194 and 195. Inside the aperture of the nostril is a slight dilation, the vestibule, bounded laterally by the ala and lateral crus of the greater alar cartilage, and medially by the medial crus of the same cartilage. It is lined by skin containing hairs and sebaceous glands, and extends as a small recess toward the apex of the nose. Each nasal cavity, above and behind the vestibule, is divided into two parts, an olfactory region consisting of the superior nasal concha and the opposed part of the septum, and a respiratory region which comprises the rest of the cavity. Lateral wall. On the lateral wall are the superior, middle, and inferior nasal conchae, and below and lateral to each concha is the corresponding nasal passage or meatus. Above the superior concha is a narrow recess, the sphenoethmoidal recess, into which the sphenoidal sinus opens. The superior meatus is a short oblique passage extending about halfway along the upper border of the middle concha. The posterior ethmoidal cells open into the front part of this meatus. The middle meatus is below and lateral to the middle concha, and is continued anteriorly into a shallow depression, situated above the vestibule and named the atrium of the middle meatus. On raising or removing the middle concha, the lateral wall of this meatus is fully displayed. On it is a rounded elevation, the bulla ethmoidalis, and below and in front of this a curved cleft, the hiatus semilinearis. The bulla ethmoidalis is caused by the bulging of the middle ethmoidal cells, which open on or immediately above it, and the size of the bulla varies with that of its contained cells. The hiatus semilinearis is bounded inferiorly by the sharp concave margin of the uncinate process of the ethmoid bone, and leads into a curved channel, the infundibulum, bounded above by the bulla ethmoidalis, and below by the lateral surface of the uncinate process of the ethmoid. The anterior ethmoidal cells open into the front part of the infundibulum, and this, in slightly over 50% of subjects, is directly continuous with the frontonasal duct or passage leading from the frontal air sinus. But when the anterior end of the uncinate process fuses with the front part of the bulla, this continuity is interrupted, and the frontonasal duct then opens directly into the anterior end of the middle meatus. Below the bulla ethmoidalis, and partly hidden by the inferior end of the uncinate process, is the ostium maxillaire, or opening from the maxillary sinus. In a frontal section, this opening is seen to be placed near the roof of the sinus. An accessory opening from the sinus is frequently present below the posterior end of the middle nasal concha. The inferior meatus is below and lateral to the inferior nasal concha. The nasolacrimal duct opens into this meatus under cover of the anterior part of the inferior concha. Medial wall. The medial wall or septum is frequently more or less deflected from the median plane, thus lessening the size of one nasal cavity and increasing that of the other. Ridges or spurs of bone growing into one or other cavity from the septum are also sometimes present. Immediately over the incisive canal at the lower edge of the cartilage of the septum, a depression, the nasopalatine recess, is seen. In the septum, close to this recess, a minute orifice may be discerned. It leads backward into a blind pouch, the rudimentary vomeronasal organ of Jacobson, which is supported by a strip of cartilage, the vomeronasal cartilage. This organ is well developed in many of the lower animals, where it apparently plays a part in the sense of smell, since it is supplied by twigs of the olfactory nerve and lined by epithelium similar to that in the olfactory region of the nose. The roof of the nasal cavity is narrow from side to side, except at its posterior part, and may be divided, from behind forward, into sphenoidal, ethmoidal, and frontonasal parts, after the bones which form it. The floor is concave from side to side, and almost horizontal antero-posteriorly. Its anterior 3/4 are formed by the palatine process of the maxilla, its posterior fourth by the horizontal process of the palatine bone. In its anteromedial part, directly over the incisive foramen, a small depression, the nasopalatine recess, is sometimes seen. It points downward and forward, and occupies the position of a canal which connected the nasal with the buccal cavity in early fetal life. The mucous membrane, membrana mucosa nasi. The nasal mucous membrane lines the nasal cavities, and is intimately adherent to the periosteum or perichondrium. It is continuous with the skin through the nares, and with the mucous membrane of the nasal part of the pharynx through the choanate. From the nasal cavity its continuity with the conjunctiva may be traced, through the nasolacrimal and lacrimal ducts, and with the frontal, ethmoidal, sphenoidal, and maxillary sinuses through the several openings in the meatuses. The mucous membrane is thickest and most vascular over the nasal conchae. It is also thick over the septum, but it is very thin in the meatuses on the floor of the nasal cavities and in the various sinuses. Owing to the thickness of the greater part of this membrane, the nasal cavities are much narrower, and the middle and inferior nasal conchae appear larger and more prominent than in the skeleton. Also the various apertures communicating with the meatuses are considerably narrowed. Structure of the mucous membrane. The epithelium covering the mucous membrane differs in its character according to the functions of the part of the nose in which it is found. In the respiratory region it is columnar and ciliated. Interspersed among the columnar cells are goblet or mucin cells, while between their bases are found similar pyramidal cells. Beneath the epithelium and its basement membrane is a fibrous layer infiltrated with lymph corpuscles, so as to form in many parts a diffuse adenoid tissue, and under this a nearly continuous layer of small and larger glands, some mucous and some serous, the ducts of which open upon the surface. In the olfactory region, the mucous membrane is yellowish in color, and the epithelial cells are columnar and non-ciliated. They are of two kinds, supporting cells and olfactory cells. The supporting cells contain oval nuclei, which are situated in the deeper parts of the cells and constitute the zone of oval nuclei. The superficial part of each cell is columnar and contains granules of yellow pigment, while its deep part is prolonged as a delicate process which ramifies and communicates with similar processes from neighboring cells, so as to form a network in the mucous membrane. Lying between the deep processes of the supporting cells are a number of bipolar nerve cells, the olfactory cells, each consisting of a small amount of granular protoplasm with a large spherical nucleus, and possessing two processes, a superficial one which runs between the columnar epithelial cells and projects on the surface of the mucous membrane as a fine hair-like process, the olfactory hair. The other or deep process runs inward, is frequently beaded, and is continued as the axon of an olfactory nerve fiber. Beneath the epithelium, and extending through the thickness of the mucous membrane, is a layer of tubular, often branched glands, the glands of Bowman, identical in structure with serous glands. The epithelial cells of the nose, fauces, and respiratory passages play an important part in the maintenance of an equable temperature by the moisture with which they keep the surface always slightly lubricated. Vessels and nerves. The arteries of the nasal cavities are the anterior and posterior ethmoidal branches of the ophthalmic, which supply the ethmoidal cells, frontal sinuses, and roof of the nose. The sphenopalatine branch of the inferior maxillary, which supplies the mucous membrane covering the conchae, the meatuses and septum. The septal branch of the superior labial of the external maxillary, the infraorbital and alveolar branches of the internal maxillary, which supply the lining membrane of the maxillary sinus, and the pharyngeal branch of the same artery, distributed to the sphenoidal sinus. The ramifications of these vessels form a close plexiform network beneath and in the substance of the mucous membrane. The veins form a close cavernous plexus beneath the mucous membrane. This plexus is especially well marked over the lower part of the septum and over the middle and inferior conchae. Some of the veins open into the sphenopalatine vein, others join the anterior facial vein, some accompany the ethmoidal arteries and end in the ophthalmic veins, and lastly, a few communicate with the veins on the orbital surface of the frontal lobe of the brain, through the foramina in the cribriform plate of the ethmoid bone. When the foramen cecum is patent, it transmits a vein to the superior sagittal sinus. The lymphatics have already been described, page 695. The nerves of ordinary sensation are the nasociliary branch of the ophthalmic, filaments from the anterior alveolar branch of the maxillary, the nerve of the pterygoid canal, the nasopalatine, the anterior palatine, and nasal branches of the sphenopalatine ganglion. The nasociliary branch of the ophthalmic distributes filaments to the forepart of the septum and lateral wall of the nasal cavity. Filaments from the anterior alveolar nerve supply the inferior meatus and inferior concha. The nerve of the pterygoid canal supplies the upper and back part of the septum and superior concha, and the upper nasal branches from the sphenopalatine ganglion have a similar distribution. The nasopalatine nerve supplies the middle of the septum. The anterior palatine nerve supplies the lower nasal branches to the middle and inferior conchae. The olfactory, the special nerve of the sense of smell, is distributed to the olfactory region. Its fibers arise from the bipolar olfactory cells and are destitute of medullary sheaths. They unite in fasciculi, which form a plexus beneath the mucous membrane, and then ascend in grooves or canals in the ethmoid bone. They pass into the skull through the foramina in the cribriform plate of the ethmoid, and enter the under surface of the olfactory bulb, in which they ramify and form synapses with the dendrites of the mitral cells. The accessory sinuses of the nose. Sinus paranasales. The accessory sinuses or air cells of the nose are the frontal, ethmoidal, sphenoidal, and maxillary. They vary in size and form in different individuals, and are lined by ciliated mucous membrane directly continuous with that of the nasal cavities. The frontal sinuses, sinus frontalis, situated behind the superciliary arches, are rarely symmetrical, and the septum between them frequently deviates to one or other side of the middle line. Their average measurements are as follows: height 3 cm, breadth 2.5 cm, depth from before backward 2.5 cm. Each opens into the anterior part of the corresponding middle meatus of the nose through the frontonasal duct, which traverses the anterior part of the labyrinth of the ethmoid. Absent at birth, they are generally fairly well developed between the 7th and 8th years, but only reach their full size after puberty. The ethmoidal air cells, cellulae ethmoidales, consist of numerous thin-walled cavities situated in the ethmoidal labyrinth and completed by the frontal, maxilla, lacrimal, sphenoidal, and palatine. They lie between the upper parts of the nasal cavities and the orbits, and are separated from these cavities by thin, bony laminae. On either side they are arranged in 3 groups, anterior, middle, and posterior. The anterior and middle groups open into the middle meatus of the nose, the former by way of the infundibulum, the latter on or above the bulla ethmoidalis. The posterior cells open into the superior meatus under cover of the superior nasal concha. Sometimes one or more opens into the sphenoidal sinus. The ethmoidal cells begin to develop during fetal life. The sphenoidal sinuses, sinus sphenoidales, contained within the body of the sphenoid, vary in size and shape. Owing to the lateral displacement of the intervening septum, they are rarely symmetrical. The following are their average measurements. Vertical height 2.2 cm, transverse breadth 2 cm, antero-posterior depth 2.2 cm. When exceptionally large, they may extend into the roots of the pterygoid processes or great wings and may invade the basilar part of the occipital bone. Each sinus communicates with the sphenoethmoidal recess by means of an aperture in the upper part of its anterior wall. They are present as minute cavities at birth, but their main development takes place after puberty. The maxillary sinus, Sinus maxillaris, antrum of Highmore. The largest of the accessory sinuses of the nose is a pyramidal cavity in the body of the maxilla. Its base is formed by the lateral wall of the nasal cavity, and its apex extends into the zygomatic process. Its roof or orbital wall is frequently ridged by the infraorbital canal, while its floor is formed by the alveolar process, and is usually 1/2 to 10 mm below the level of the floor of the nose. Projecting into the floor are several conical elevations corresponding with the roots of the first and second molar teeth, and in some cases the floor is perforated by one or more of these roots. The size of the sinus varies in different skulls, and even on the two sides of the same skull. The adult capacity varies from 9.5 cubic centimeters to 20 cubic centimeters, average about 14.75 cubic centimeters. The following measurements are those of an average-sized sinus. Vertical height opposite the first molar tooth, 3.75 centimeters. Transverse breadth, 2.5 centimeters. Antero-posterior depth, 3 centimeters. In the antero-superior part of its base is an opening through which it communicates with the lower part of the hiatus semilunaris. A second orifice is frequently seen in or immediately behind the hiatus. The maxillary sinus appears as a shallow groove on the medial surface of the bone about the 4th month of fetal life, but does not reach its full size until after the second dentition. Footnote: the various measurements of the accessory sinuses of the nose are based on those given by Aldrin Turner in his Accessory Sinuses of the Nose. End of footnote. At birth it measures about 7 mm in the dorso-ventral direction, and at 20 months about 20 mm. End of section 48.